Don't be nervous about that part. So no, that'll be fun. So my daughter did a really mean thing this week. She called up and she said, guess what I'm doing this weekend? I'm going skydiving. It's like not exactly what your parents want to hear, right? And I said, I always told you, don't tell me if you do that. Tell me afterwards that you do it, so I don't worry. And she said, oh, don't worry. We got a really good coupon, and so it's really cheap. Like, oh, this just gets better and better. Budget skydiving. Okay. So a little worried about that for this weekend, but hopefully she'll get through it. I did post this morning a few announcements that I just wanted to reiterate in class about our exam on Tuesday. So same length as the first exam, 62 questions. We'll cover chapter 5, chapter 7, and through today. I'm hoping to get through the viruses today. We're not going to go on to any new material today. Hopefully that will come out about right. I would like as many of you as possible to take it in person. Email me if you have some situation. I will make it available by Respondus, but you're going to need a password to do it by Respondus. So you need to email me and talk to me if you want to do it that way. I would like most of you to take it in person. So just be aware of that. Let's see. I will try to get a few practice questions up either today or tomorrow, just like last time. 10 or so practice questions, just kind of see where you're at maybe. So also homework is due Tuesday. I really think this homework for chapter 5, especially for the genetic stuff, is really quite good. So don't blow through that in as little time as possible. Spend some time with that, watch the animations, get it out of it. I think it will be helpful for you to, again, spend a little time there. So don't wait till the last second to do that homework. All right, when we left off last time, I had hit the wrong button somewhere and had a little trouble. What I wanted to write on this slide, my last point there, was that envelope viruses are a little bit wimpier in the environment. They typically last maybe only hours, up to maybe a couple days. It depends on the virus, it depends how hardy they are, but that envelope, phospholipid membrane envelope dries out really easily. And so doesn't afford, and then if that envelope dries out, then the virus can't recognize the host cells in order to, attach to those host cells and eventually gain entry. So that really renders the virus not viable anymore if its membrane is broken down. So we were continuing on with just some kind of general characteristics of viruses. We'll get into viral replication today and end up with another small infectious particle, prions. All right, so we talked about, actually, let me go back to this slide again. At the end of last time, that a lot of viruses have these spikes on their surface. Sometimes we call those peplomeres, but you can see the spikes on the influenza virus. The adenovirus has some coming out of its capsid. Not all viruses have spikes, but if they don't have spikes, they have some proteins embedded in their capsid that would allow them to recognize certain kinds of cells. So viruses are typically pretty specific in that they only will infect a certain host or even certain cells within a host. HIV, pictured right here in, that's the little pink circles, is human immunodeficiency virus. That virus is specific for several types of cells in our body. The most famous one is the CD4 helper cells that it infects. It infects some other cells and just doesn't harm them like it does our T cells, our T helper cells. So that here's the HIV, and the whole background here is a white blood cell, is a T helper cell. If we swallow HIV or inhale HIV, that doesn't make us sick. Okay, so it needs a specific kind of cell even within, a specific host. viruses that infect bacteria, here this E. coli or whatever bacterial cell that is definitely in trouble. Lots of bacteriophage trying to infect that cell. They're specific usually for one particular species. So really specific. And of course, there are viruses that infect plants as well. This is tobacco plants infected by a tobacco mosaic virus. So we have viruses that infect animals, bacteria, plants. We think that there are viruses that infect fungi, but the fungal cell wall is so thick and tough that typically it's only passed between fungi when their hyphae fuse with each other. Okay, so a little bit difficult to pass between organisms, apparently. And there are some viruses that are more generalists that are not quite as specific, like West Nile virus infects lots of species of birds, mammals, us as well. So often they're specific, but not always. All right. The genetic material of virus, we've kind of mentioned this in passing already, that all viruses, they all have some kind of nucleic acid. They either have DNA or RNA. The DNA could be double-stranded DNA. It could be single-stranded DNA, which is a form of DNA that's obviously never found in living or other living organisms. Well, we'll talk about whether we consider viruses living or not. The RNA can be double-stranded RNA, which again, never found in our bodies. We never have double-stranded DNA or single-stranded DNA. RNA. Okay. And that nucleic acid can be either circular or linear. Circular, kind of like a little plasmid, or linear. And the linear we can describe as being segmented or unsegmented. So here's an example of this virion up here that has a segmented genome. I'll draw like this. Okay, it's got 3 different segments of whatever kind of nucleic acid that is. If it was unsegmented, it would just have, you know, one long line of nucleic acid. Well, what's the difference? Why would a cell have one kind instead of the other? A lot of viruses, especially RNA viruses, mutate really quickly because the enzymes that they use are pretty indiscriminate and they make mistakes often when they're replicating their, well, copying their DNA or RNA, I'll say. So if it's a virus that mutates a lot, if it's got a segmented genome and there's a mutation, you know, that causes all of these gene products, you know, not to function, then possibly this virion could still be viable because it's got, you know, encoding for other proteins that would maybe work fine. If it had its genome all in one segment and had a big mutation right here, all the rest of it, you know, wouldn't be copied or whatever, and probably the virion wouldn't be viable to infect another cell. Okay, the gene number is always small for viruses. How small is it? Well, if you compare it to humans, after we completed the human genome project and sequenced all of our DNA, we realized that actually the human genome is much smaller than what we had anticipated. We thought we had over 100,000 genes. We're still not exactly sure. You can't exactly tell from the sequence of DNA what makes a gene and what doesn't. But we now estimate humans have between 25,000 and 40,000 genes. Compare that to a bacteria like E. coli has about an order of magnitude less in terms of number of genes, about 4,000. And although there's some variation between viruses, for instance, herpes virus has between 100 and 200 genes. The virus with the smallest genome is the hepatitis B virus with only 4 genes. And when you think about it, that makes sense. And a little analogy that I use for that, or that dawned on me last year, bear with me, when my son came home from fall break, we went and picked him up at his friend's house. He got a ride. He jumped in the car and he said, okay, ready to go. And I said, well, wait a minute, where's all your stuff? He had a backpack with him that had his computer and a football. He said, no, this is all I brought. This is all I need. I said, you're gonna be home for 4 days. And he said, well, I don't need anything, you know? And I got to thinking, you know, after I did my mom's sigh, that he was right, because at home, you know, we had a bed for him, we had towels, we had all the food he wanted. He had some clothes that were old that maybe still fit him. We had an old toothbrush. Everything that he needed really was at home already. So same thing with viruses. They come and they gain access to living cells and they just take over that cell and use everything that that cell has already. There's maybe only a few things that they need specifically to make their own viral proteins. So it makes sense that they carry a very, that they travel light. We'll put it that way. So I call my son my hepatitis B virus sometimes. He loves it. Okay. So you can actually view the genome of cells. I'm not quite sure how this is prepared, but here's a partial genome of E. coli. And right up here, you can tell that somehow they can tell that E. coli has been infected with some virus, and that viral genome comparatively way smaller than the E. coli genome. What these genes that viruses bring in with them, what are sort of the functions that they might need to bring with them that the cell doesn't already have? They might need to produce some of their own proteins, like their viral spikes. They express them on the surface of the cell that they are infecting, and then they grab them as they go. Maybe some kind of enzymes or proteins to regulate the host cell in some way. Most importantly is they have to be able to replicate their own genetic material. So sometimes I cover viruses before we've done genetics. So I like to remind students, remember, when DNA is copied, we call that replication. When messenger RNA is made, we call that transcription. And when proteins are made, that's translation. You all know that, of course. But if the virus, say, is a RNA virus, it needs enzymes to copy its own RNA. We don't have enzymes to do that. We never do that. So that's what replicase is. Typically, those are enzymes that copy RNA. It might need some specific polymerases. You know, it might bring its own copy of a DNA polymerase to make messenger RNA. Some viruses need reverse transcriptase. That's an enzyme that copies DNA from an RNA template. Okay, when that was discovered in 1970, that was really a mind-blowing thing, because obviously that never happens in our cells. We never make DNA from RNA. Okay, that was David Baltimore was one of the people that discovered that. Okay, and I mention him because he actually has formulated one of our schemes for classifying viruses. This is considered, we have two different viral classification systems. This is what we would call the Baltimore classification. Okay, and this is pretty straightforward. He has classified all viruses based on their genome. So 7 classes, Roman numerals there. Not too much arguing there about where viruses belong. So there's double-stranded viruses, like I mentioned before, single-stranded DNA viruses, double-stranded RNA, 2 different kinds of single-stranded RNA, and I'm going to talk shortly about the difference between plus sense and minus sense. Try to illustrate that a little bit better. The retroviruses, those are the ones that have the reverse transcriptase that basically start out single-stranded messenger RNA. This is like HIV, it's one of these viruses, and then make DNA from that, incorporate their viral DNA into our DNA. Okay, but we'll talk more about HIV later. And then there's this one small group of viruses, which actually hepatitis B happens to be a virus that copies their RNA through a DNA intermediate. So again, viruses do lots of crazy stuff that we don't do in our cells. The other more traditional way that we have classified viruses, the Baltimore classification has been fairly recent. We refer to it as the hierarchical. I knew I would have trouble spelling this. Hierarchical. No E here. There you go. Forget that E. Hierarchical classification. With this classification, we kind of act like viruses are living cells. And how would we classify them if we considered them living? So we try to kind of force them into our same system as all other life. Only we don't have kingdoms and phylums really for viruses. The biggest, there's 3 orders of viruses that have been described. And then below that, there's families. Viral families end in viridae. So herpes viridae is the family that herpes virus fits into. It's grammatically correct to italicize family names, just FYI. And then there are genera that have the suffix virus. So herpes virus is in the family herpes viridae. Species is a little bit more loosey-goosey. Oftentimes we say the species is the disease that the virus causes. Like chickenpox, that would be considered the species that the herpes virus causes. Okay, and what do we use to put those, put viruses into these genus and families? We look at the characteristics of which we have been talking about last lecture and this morning. What kind of nucleic acid is there? The symmetry of the capsid. Is it helical? Is it icosahedral? Does it have an envelope? Maybe to some extent, how big it is. Okay, those are all things that then we use to classify in that hierarchical classification. And I just included this picture mostly just to kind of appreciate the diversity a little bit. It's probably too small to see up on the screen for sure. But again, these are all things we've talked about. The nucleic acid, symmetry of capsid, is it naked or enveloped? Here's the Baltimore class included here. The genome architecture, we talked about whether it's segmented. Here it says continuous instead of unsegmented. Okay, what the DNA looks like. It's got family names here. So again, don't memorize anything specific from that. Just it's kind of a nice all-inclusive thing. We've got Corona family there. All right, so let's get in a little bit more detail. There are 2 main ways that viruses replicate. We're going to start with the lytic replication cycle that all viruses can do. Some of them then go into this other replication cycle where they can actually integrate into the host chromosome. Okay. So a virus is only going to replicate when it's in a living cell. All right, and usually it's going to kill that host cell. Not always right away, but eventually it typically will. So there's 5 stages that we'll look at, and we'll go through them one by one. Attachment, entry, synthesis of new virus parts, assembly of all this together, and then how does it release out of the host cell. And we're going to talk first about viral replication in bacteriophage. So bacteriophage, remember, are viruses that infect bacteria only. So here's lytic replication of a T4 bacteriophage. This is a double-stranded DNA virus. Most bacteriophages are DNA. There are a few exceptions. Most of them don't have envelopes, so most of them are naked. All right, and they have this, again, weird, I showed you one of these last time, weird shape to them, looks kind of like a lunar lander, but these bacteriophage land on a host cell, and they don't have any way to move, right? There's no flagella, anything like this, so just by kind of random motion, they'll land on a host cell that they are specific for. There are, in these tail fibers, there are proteins that are specific for receptors on the outside of E. coli. So this is actually a bacteriophage that only infects E. coli. Okay, so you can see this is gram-negative. There's an outer membrane and inner membrane there. Okay, so attachment, it lands on and recognizes that E. coli cell. When it enters, that means that the nucleic acid is somehow injected into the cell. And it kind of looks like the tail fiber there kind of contracts a little bit. It almost looks like a little syringe shooting that nucleic acid right into the cell. It actually needs a little bit of help from an enzyme that it carries with it, lysozyme. Lysozyme is an enzyme that breaks the bonds between the sugar subunits of peptidoglycan. And so it weakens the cell wall. Okay, so that typically is necessary to weaken the cell wall a little bit in order to get that nucleic acid inside of the cell. Okay, so the only thing that goes inside the cell is that nucleic acid. The whole rest of this phage basically just falls away. It's an empty shell. The phage DNA carries with it enzymes to degrade the host chromosome, because that bacteria now wants, or sorry, that virus wants to be in charge instead of the host DNA being in charge. It will take over the cell's machinery. It will, you know, make messenger RNA using the cell's enzymes and make its own proteins. So that's the synthesis phase, where again, it's making its own viral proteins. And then in assembly, it's taking all those parts, all those proteins that it's made. And if you kind of think assembly line at GM, basically they just all come together. There's not enzymes that mediate that process. We think that they just spontaneously come together somehow. That's sort of hard to wrap your mind around. And then once you have all these viruses assembled there, then they're going to lyse the cell, be released. And that also involves the lysozyme to weaken the cell wall so that the new virions can burst out. All right, whole process takes about 25 minutes. All right, so pretty quick. All right, if you look at a graph of the numbers of, when we compare this to like a growth curve that we'd looked at for bacteria, it only takes one virus to enter a cell. And then after about 25 minutes, we get a burst, the cell lyses, and somewhere between 100 to 200 new bacteriophage are going to burst out of that bacterial cell. Okay, so very abruptly, right? You gotta almost feel sorry for this E. coli here. Nice picture caught in the process of it bursting open, releasing lots of new bacteriophage. Okay, and I just stumbled upon this newspaper article. I didn't post this, I don't think. This actually was in 2016. And I love it when in the newspaper they try to explain something scientific. Some things they get right and some things are really not quite so right. But we have explored the use of bacteriophage in order to kill bacterial infections for almost 100 years now. In this country, we kind of quit exploring that so much when we came out with antibiotics. We thought antibiotics were our savior. We didn't need viruses to kill bacteria anymore. We just had our own drugs. Of course, now antibiotics, there's lots of resistance, other possible problems with using antibiotics. So we're getting more and more interested in using specific viruses to kill off bacteria that are causing infection in us. And there's actually more research on this going on overseas. But basically this group in Paris isolated viruses from a sewer, and then they tried to use these bacteriophage. They found some that were specific for a bacteria called Pseudomonas, which can often cause hospital-acquired infections, especially in burn victims. Okay, their skin's burned off, they've lost their first line of defense against bacterial infections. So they soaked bandages in this bacteriophage solution and put these bandages on burn victims. And they did a really good job. They were much less likely to get a bacterial infection. So I love the way they explain how this works here. The bacteriophage parachutes down onto a bacterium. It uses its sharp tail to poke into the bacterium and injects its genetic material. Baby phages take over the bacterium's body. There's not really baby phage or adult phage, they're just assembled, remember, but that's kind of cute. And then the phages burst out and propagate. So definitely an interesting area of research. Okay. So the second way that viruses can replicate is called the lysogenic replication cycle in bacteriophage. And this here example organism we've used is called lambdaphage. And that we consider phages that can do a lysogenic replication cycle, we call them a temperate. It's a temperate phage. Okay, I forgot to use that term before. A virulent phage is one that can only do the lytic cycle. Okay, so temperate phage is able to also do lysogeny or a lysogenic replication cycle. Okay, and here's a electron micrograph of a lambdaphage down there. You can see that they don't have the tail fibers that the T4 phage did, but it's got those proteins at the tip of the tail that recognize, I believe this is another phage that only recognizes E. coli. So you can see the attachment phase here. Entry, again, that lysozyme is necessary to weaken the cell wall a little bit. But instead of just breaking up the host chromosome, instead, here's the viral DNA. Instead, it incorporates into the host chromosome. So we call this a prophage. Just circle this. This is the prophage, when the virus has incorporated into the host chromosome. Okay, so I left this little bit written over here. Virus DNA incorporates into host DNA. And that bacteria that's infected then basically can carry that lambdaphage with it. It will replicate every time the cell replicates. So sometimes for generations, the cell will replicate and have that prophage there within its chromosome. So It doesn't kill the cell right away. It just is there over generations. Sometimes we can get what's called a lysogenic conversion. And that means this bacteria expresses certain proteins because of that virus that it didn't before. So the bacteria actually can change its phenotype due to that bacteriophage DNA. So we know there are some examples of this. For instance, cholera, it only actually infects humans if that bacteria has been infected with a bacteriophage. There's some streptococcus that would cause scarlet fever, only cause scarlet fever if it's been infected by a phage. Diphtheria only makes diphtheria toxin if it's been infected by a phage. So we know that again, that change in phenotype, that lysogenic conversion takes place or can take place with certain organisms. At some point though, that phage DNA can come out of its host cell, and we call that induction. Okay, so I'm going to use this abbreviation for phage. We'll circle with a stick through it. Phage comes out of host chromosome. We're not sure, host chromosome. We're not sure why it does that. We know that if the cell is stressed in some way, induction will happen. We can make it happen by like putting radiation on cells. So if the cell is going under some stress, basically the virus jumps ship, recognizes, hey, I need to get out of here. I gotta make more of myself. So then it enters into the lytic cycle where it's making its own proteins. We'll assemble them and then be released. Okay. Oh, and I should mention that the lambda phage also encodes for proteins so that you can only get infection by one lambda phage at once. So it suppresses infection by other phage. Okay, because if you think about it, if you've got 100 lambda phage infecting this cell all at once, and they're all integrating into that host cell chromosome, that cell is probably not going to be viable anymore. So really only want one at a time. So once one is infected, it somehow signals to other phage, and they're not able to infect the cell also. Okay. All right, so let's expand our view now to think about viruses that infect animals. All right, so we had two examples of bacteriophage. Replication for animal viruses is much the same with a few differences. So same basic replication pathway as bacteriophage. There's differences because many animal viruses affect us, humans, are envelopes. So that makes it a little bit different how those envelope viruses enter cells and how they leave, especially. The eukaryotic nature of animal cells, that's a really kind of mysterious way of just saying, remember, our cells have nuclei. And if the viruses are going to replicate their DNA, they usually have to go where our DNA is, because that's where the enzymes are that they need. So they typically have to go to the nuclei, not always. And of course, animal cells, what do they lack? They lack cell walls. Okay, so again, that makes them able to endocytose and exocytose. Lots of differences, again, with entry and release. Okay, so if we look at 3 mechanisms of entry, we're going to actually think both about bacteriophage and animal viruses here. But the 3 ways that the virus gain access to a host cell. Again, these pictures are from your book here. Direct penetration. So here, it actually shows a picture of a phage that's just basically a capsid. It doesn't have an envelope or any tail fibers or anything. But that phage is basically recognizing receptors on the cytoplasmic membrane of an animal cell, and it attaches, and then basically a pore forms there, and the genome is basically enters the host cell. So very simply, we call that direct penetration. And this is going to be naked viruses only. Okay, so by naked, I mean viruses that don't have an envelope. Membrane fusion. Obviously, this method is enveloped. That's an O, enveloped viruses only. Okay, so here's a virus with an envelope around it. With this method, the envelope basically fuses with the cell membrane. Here's the capsid that becomes uncoated or falls away once it's inside of the cell, and then basically you're left with that viral genome floating around. A third method, endocytosis, this can be naked or enveloped viruses. Okay, method of entry. So with this method, some signal is made from the virus to the host cell to get the host cell to envelop the virus, endocytose it, and then once that virion is inside of the cell, somehow the membrane falls away, capsid falls away, and eventually you get the virus, okay? So one big advantage with endocytosis for the virus over membrane fusion is with membrane fusion, you can see here, there are viral glycoproteins remaining in the host's cell membrane. That is a big red flag to that host immune cells. This cell is infected with a virus. So it's kind of an advertisement to our immune system that there's a problem there. Now we're gonna look later on at how our immune system would know that the cell is infected with a virus, even if that advertisement isn't left there. But it's an early signal that that cell isn't infected, and maybe the virus wants to be a little stealthier than that. Okay, so by one of those methods, that viral genome has gained access to the cell. Really, the first order of business is to copy that genome so that the virus can make more of itself. And again, it depends on what kind of nucleic acid that cell has to make more of itself. So, we've talked about this a couple times now, how there's a lot of variety here. I want to look at the single-stranded RNA viruses a little bit more carefully. A positive sense RNA virus, their genome basically acts as messenger RNA. Okay, that's in a form that's ready to be read by the ribosomes. Okay, if it's a negative sense RNA virus, then it has to be copied to a positive sense RNA before it can be read at the ribosomes, translated. Okay, so just to illustrate that, let's say the messenger RNA is, here's the beginning of the sequence of it. Okay, so here the AUG, that's a codon, ready to be read at the ribosome. CCC, that codon actually corresponds to the amino acid proline, so that's ready to go, ready to be read. The complementary strand there would be UACGGG, right? UAC codes for tyrosine and GGG for glycine. Okay, if that shows up at the ribosome, that's not a sequence it's not gonna make any sense. Okay, it has to be copied into the positive sense strand. Okay. As a rule, DNA viruses have to have access to the nucleus because that's where the enzymes are that the host has to make RNA from DNA. There was one exception there. Smallpox virus is a DNA virus that only replicates in the cytoplasm. It just brings its enzymes with it. So this is one exception. RNA viruses don't need to go into the nucleus or anything because they're going to have everything they need, the enzymes that they need are going to be out in the cytoplasm. Okay. All right, so this is your book's graphic, just thinking about how those genomes get replicated. Of course, if it's a double-stranded DNA virus, then it gets replicated just like our DNA does. But if it's a positive sense single-stranded RNA virus, example of that, coronavirus, again, it's just in the form that it needs, it can be read right at the ribosome, it can be translated. Also has to make more of itself, so it does that through an intermediate of negative single-stranded RNA, and that negative strand is copied to that single-stranded positive strand. Okay, if you have a negative sense single-stranded virus, there's lots of these. Flu virus is one, influenza virus, Ebola is another one. Basically, that has to be made, again, into positive sense single-stranded RNA. That can be used right there at the ribosome and copied into negative strand to be assembled. Double-stranded RNA viruses, these are pretty rare. The only example I could think of, and again, don't worry about memorizing that, Rioviridae, that's a virus that's rotavirus, so causes bad diarrhea in usually babies. A double-stranded DNA virus would have to unwind. Okay, the positive sense, again, can be translated. Both the positive and the negative sense are copied to make that double-stranded. So again, just a little bit different strategies depending on what kind of nucleic acid it is. But, uh-oh, bumped something there. Come back. Hopefully, I was afraid it would do that. Let's give it a second, see if it comes back. Okay, come back. Good. Okay. No matter what kind of nucleic acid is here, the point that I'm trying to hit home here is that it has to be able to make positive sense RNA. Okay, so again, don't memorize all these virus names, but whatever kind of nucleic acid it has, it's got to make messenger RNA, positive sense, single-stranded RNA, because that's what our ribosomes would recognize. Okay, so that's just the point of that. All right, so once everything is synthesized for the virus, assembly and release. So we talked about the bacteriophage. The time that it took to replicate was about 25 minutes. That's pretty common for a bacteriophage. For viruses that infect animals, they typically take a lot longer, about 24 hours, okay? So minutes versus hours there. Okay. And again, that bacteriophage, about 25 minutes. So just to compare there. Your book doesn't include a very good picture here for release, But if the virus is naked, if it doesn't have an envelope, it can be released by two different ways, by exocytosis or lysis. Okay. With exocytosis here, just the virus basically travels in a membrane-bound vesicle and then is released. The virus doesn't acquire any of the host membrane. Okay, it doesn't need it. That's the opposite of endocytosis, right? Exocytosis. And then enveloped virions are usually, they can also be released by exocytosis or budding. So if they're being released by exocytosis, then the virus acquired the membrane from somewhere else other than the cell membrane, like the nuclear membrane. Herpes virus does that. It gets its envelope as it leaves the nucleus, takes some of the nuclear membrane with it. And then it just travels in a membrane-bound vesicle and gets released. That's exocytosis. Or budding is when the virus takes some of the cell membrane. So the next slide is a nice graphic of budding. And basically that viral capsid, it typically would express the proteins that it needs into the host cell membrane here. And then as it leaves, as that capsid leaves, it just takes some of that host cell membrane with it. Okay, so that's budding. If you look at a sort of a growth curve, we'll call it, viruses that do budding, it's more of a slow release kind of over time. Compare that with that rapid burst that we would get during lytic replication when, you know, a bunch of bacteriophage burst out all at once. It's generally a slower release, but if lots of virions are leaving by budding all at once, it can basically have the same effect as a burst. Basically the cell membrane, there's not enough of it left for the cell to be viable, and it can still kill the cell pretty quickly. It happens slower than not so much. You might have more of a persistent or chronic infection. Okay. All right, so looking at lysogeny in bacteriophage, viruses that infect animals basically do a lysogenic cycle, but we call it by a different name. Okay, so we call it latency when animal viruses incorporate into the host cell chromosome. Oops. There we go. Okay, so animal viruses can remain dormant in their host cells. HIV can do this. Herpes virus can do this. People of my generation that had chickenpox when they were young, that virus can reactivate later in life in an infection known as shingles, because that chickenpox, that herpes virus, never actually went away. It's incorporated into the chromosome of people that have had it. So viruses can definitely stay dormant. Just like in lysogeny, how the bacteria cell can carry with it for many generations, this may last for years with no virus activity. It should say activity. It's hard to read my writing there. Okay, so that, you know, virus can just be hanging out. But the difference between bacteriophage and animal virus is that we don't get induction for animal viruses. Once that virus chromosome is incorporated into our chromosomes, as far as we know, it's there forever after, okay? It doesn't ever leave and then enter any kind of a lytic cycle, okay? So I just posted that quickly there, just a reminder, we just did that though, of the lytic cycle. I want to point out a few differences. Remember, this was the prophase here. In bacteriophage, basically we get the same sort of thing for animal viruses, but we call that stage a provirus stage, or we call it prophage. Okay, and that's again when it's incorporated into the host chromosome. Okay, we just have, again, a little different terminology for it there. And like I said, we can get induction when the virus leaves the host chromosome. But for animal viruses, we get no induction. Okay, like I said, once incorporated, it's always going to be there. Okay, I think we're doing okay. All right, so how do we grow up viruses? I had some of my students in lab, swabbing places in lab and, spreading it on an agar plate to see what grows up. We could get some fungus growing up when you do something like that. You could get some bacteria, but we're not going to get viruses growing on that media. Remember, viruses need growing living cells in order to replicate themselves. So if we were going to try to culture viruses, we could do that in living bacteria. You would have to make a lawn of bacteria on an agar plate and get those, give those cells some time to grow up, to get confluent growth on there. And then if you add a suspension of viruses, usually you would dilute that out, maybe lots of different dilutions to try to get kind of an appropriate number. And then usually you put another overlay on there just so that the bacteriophage don't kind of replicate too quickly. But the bacterial cells that the bacteriophage infect will lyse and die, and eventually that happens with more and more neighboring cells, and you get little plaques showing up. We call this a plaque assay. And so you're looking for areas where the bacterial cells have died. And you can count those plaques and then extrapolate to about how many phage you had in your original solution. It's kind of the opposite of the viable plate count that we talked about a little while ago. Okay. So again, allows estimation of numbers there a little bit. So we used to do a lab like this in 213, but it wasn't working reliably, so we kind of gave up on it. Okay, other ways we grow viruses. It's very common to grow viruses in eggs, especially viruses that you want to make a vaccine for. It depends on the virus as to where you would inject into the embryo, an embryonated egg, an egg that's been fertilized, where it will grow best. It depends on the virus, but this is a way that we make up influenza vaccine. And of course, this takes quite a lot of time. So I know my daughter said where she lives, they're out of flu shot and they might not get any in for a while. Again, it just takes a while to manufacture. Another very common way to grow viruses, of course, you can grow them in living animals. That's the most expensive way to do it. Eggs are a little bit cheaper. Cell culture is a little bit cheaper than using eggs. So that's what I mean by less expensive than growing virus in animals or eggs. We call this cell culture. Kind of old name that we used to use was tissue culture. We should call it cell culture now. The big problem that you always have with trying to grow viruses in cells is that it gets very easily contaminated with bacteria. So you usually add some kind of antibiotic to make sure that bacteria don't contaminate it. You can grow it in these bags like this, or more often some kind of plate with individual wells in it. You see it. Use some kind of cells. Diploid cells are animal or plant cells that are prepared from embryonic cells. They usually last about 100 generations before they would die. It seems like a lot, but I guess a generation can be pretty short. If you use continuous culture, you're going to use some kind of cells that have been derived from a cancerous cell, from a tumor cell. And then they live indefinitely. Okay, so there's no limits on the number of generations there. HeLa cells are the kind of the famous ones. They've been taken from Henrietta Lacks, somebody that died of uterine cancer. I think it was might have been cervical cancer, forget. All right, so that's a little bit how we grow up viruses. Kind of to wrap up our discussion on viruses, we like to think about, well, are viruses considered alive, or are they not considered a living cell? This is something that scientists will probably argue about for a long time. It's not really settled, but if you look at the characteristics of a living organism, like living organisms, they grow, they reproduce, they are responsive, meaning they change their behavior depending on the environment that they're in. They have metabolism, they take in nutrients and make energy. So do viruses fit? Do they satisfy those requirements? Viruses do not grow, right? Like I said before, you don't have baby viruses that grow up into adult viruses. They are just assembled. Do they reproduce? Yes, they do that, but they must be in a living cell. Now, there are some bacteria that have to be in a living cell as well. So that's not only a virus thing. The responsiveness question. This is often what people will argue about the most. And I would argue no, but they do evolve over time. So you could say, well, that's maybe responsiveness. They do evade host mechanisms or host defense mechanisms. So you could maybe call that a behavior if you were stretching it a little bit. But in general, they don't respond to stimuli. So you can't, shine a light and, oh, if you shine a light on them, then they do this. Like, they don't change their behavior in response to any kind of stimulus. So that's one that people like to argue about, but most people would say, no, they don't really do that. Do they have metabolism? Definitely not, right? They don't eat. They don't absorb any kind of nutrients. They just kind of are there. So most people would argue that they are not alive. All right, so I want to wrap up our chapter 13 here with a little discussion on prions. Your book also has a little blurb about viroids. Viroids are little pieces of RNA that infect plants. So we're not really going to think any more about those. Prions, that name was coined by Stanley Prusiner in 1982. And he called them prions as being short for proteinaceous infectious particles. Okay. So this was another one of those really mind-blowing things when we understood what prions were, because they do not have nucleic acid. Okay, so that really violates the universal rule that organisms, have to make proteins from messenger RNA. Okay, they don't do that. So their protein, so the question is, how do these proteins replicate if they don't have nucleic acid? Well, here's what we know about them. PRP, this stands for prion protein. And this is a protein that basically all mammalian cells has this protein that we now call prion protein. We don't really know what function that protein has in our cells, just that we have it. And when a regular or we call it cellular PRP, when a normal version of that protein is exposed to the prion form, which is folded a little bit differently. It's got, you know, it looks subtle, the difference between these two in this picture, but the prion PRP has more of these beta sheets, apparently, rather than alpha helices, so it's folded differently. And when a normal version of PRP is exposed to prion PRP, then the normal version gets misfolded. Okay, so exposure to the misfolded PRP causes normal PRP to misfold. All right, so basically that prion PRP is a bad influence on the normal PRP. And well, when it misfolds, basically it tends to clump together, and eventually that keeps neurons from conducting nerve impulses. The clumps form kind of these plaques. So you can see in this picture here, again from your book, here's some normal brain tissue, and here's some brain tissue that's got some prion exposure. You see these white plaques where, again, the neurons aren't functioning properly. So if you think about some examples of disease caused by prions, what they all have in common is that they are all fatal. Okay, they will all kill you or the animal or whatever we're talking about. Your book talks about, oh, there's some new, you know, new therapies being formulated. Well, they talk about that, but there's not anything that's been approved. They think they've got some therapies that might slow the progress of prion disease, but there's nothing been approved by the FDA that works yet. So anyway, we're working on it. So some examples of prion disease. Probably the most famous one is the mad cow. So early 90s in England, they had lots of cows that were acting weirdly. They were acting like they were drunk almost. And so they called this bovine spongiform encephalopathy. Can't speak very well. I was in England in the early 90s, and they kept saying, you know, to our group, don't eat hamburger, don't eat any beef while you're over here. And I thought, well, that's crazy. It's cooked. How can I be getting something infectious? It's really hard to kill prions. You have to heat them at like 400 degrees Celsius for like 4 hours to deactivate them. So just cooking some meat that was infected with mad cow wouldn't be enough to inactivate these prions. So when people ate infected meat, the disease that we call in humans is variant Creutzfeldt-Jakob. So basically we'll say mad cow in humans, or caused by eating contaminated beef. Okay. And there's about 200 people that died, again, I think mostly early 90s, from variant Creutzfeldt-Jakob. Creutzfeldt-Jakob disease, it's a little complicated. So variant is when you eat contaminated meat. Creutzfeldt-Jakob disease is just kind of the genetic form. So anybody could get Creutzfeldt-Jakob if you had some kind of mutation arise. Very rare, but it's been described. Scrapie is a prion disease in sheep. It causes them to act very oddly. They scrape up against things, scrape their wool off, so that's why they call it scrapie. Chronic wasting disease is found in deer, elk, and moose, I believe. So you hear about cases every once in a while in Michigan of deer with chronic wasting disease. As far as we know, that can't be passed to humans. We haven't found any cases of humans getting prion disease from eating infected meat. But all these diseases, there's a few more. Kuru is the one that they think has been eradicated now, but there was a tribe in New Guinea, I think, that it was their ritual out of respect for their dead relatives to eat all parts of their bodies, including their brains. So we call it cannibalism, but it was more of a respectful of somebody that was already dead. Well, they've been taught not to do that, and so now they don't have Kuru anymore. There's another one, fatal familial insomnia, that's, again, very, very rare, but it runs in families. So prion disease can be inherited. It can be just some kind of sporadic mutation like the CJD, or infectious, again, like eating some, you know, meat from a cow that was infected. Okay, and it used to be our practice to grind up animals and put that ground up animals into animal feed. So basically we were cows were passing it to each other because they would eat ground up cow brains or ground up sheep brains, right? They've outlawed that now. You can't use dead animals in animal feed to try to slow down, you know, the passage of prion disease. All right, any questions about that? So that's kind of the highlights. We have a few minutes left. I've got a few practice questions if you're interested. How much time do we have left? 7 minutes. Okay, so if you want, I've got a few practice questions. Most of these are about the genetics because, like I said, it gets confusing. Kind of thinking about the differences between transcription and translation. All right, the site within the ribosome that holds the growing peptide, sometimes there's a little confusion about this. The new peptides, the tRNAs that are carrying amino acids, the new ones come into the A site, except the very first one, the formylmethionine, comes into the P site. And it's going to be that P site, that's the one in the middle that holds the growing peptide. Okay. P stands for peptidyl. I wasn't sure, I kind of mentioned this again today in lecture, but I wasn't sure if I really mentioned this very clearly or not. In bacterial cells, where does transcription take place? So you have to think about where's the DNA in bacterial cells and where the RNA gets copied. Okay, that's going to be taking place in the cytoplasm. Right, eukaryotic cells, the transcription is going to be where the DNA is, which would be in the nucleus. And although we have special regions in bacteria cells, kind of where the DNA is, it's not membrane bound, so we just still consider it cytoplasm. All right, and one more. Okay, I don't know where this line came from, ignore that. What type of mutation would you expect to have the greatest effect on a protein function? I like this question. We talked about silent mutations, missense mutations, and nonsense mutations. I'll just give you a second to look at that. Okay, so obviously if it was a silent mutation, that's not going to have any effect on the protein, so you can rule that out. Usually nonsense ones are the worst because those are the ones that encode for a stop codon instead of amino acid. But in this case, it's at the very end of the protein anyway. And same thing with the missense, it's encoding for the wrong amino acid. You know, it's at the very end. That may or may not have an effect. If you had some kind of insertion or deletion that was causing a frame shift, a change in the reading frame near the beginning of the protein, that is most likely, it's going to change the whole rest of the protein. So most likely that would be the worst one for the cell there. Okay, if you got any questions, hang around, or I have office hours. Otherwise, I will see you on Tuesday.